We left off looking at the different phases of the action potential, and then we are going to pick up here speaking about some of the general principles of the action potential and kind of sort of taking a step back and zooming out and looking at the action potential more broadly. Like, what is its function? How does it communicate? And what are some principles that govern the action potential that differ from what we said about graded potentials in general? So the all or none principle. This is basically the idea that an action potential is either going to happen in totality or not at all. So that threshold that we spoke about, that value is really important because once the membrane potential of the cell supersedes or passes that negative 55 value, there's no turning back, right? It's going to always result in a complete action potential as long as we get to that threshold value. And so a threshold depolarization always results in an action potential. A sub-threshold depolarization, anything less than negative 55. If it's negative 54.5, it's not going to result in an action potential, right? So really that threshold value is super important in terms of whether this occurs or not. If we have a supra-threshold depolarization, it's going to be the exact same action potential. So all action potentials have the same amplitude in terms of the horizontal space on the curve, right? It's always going to be the same amount of time. It's always going to have the same duration. And it's always going to go to the same membrane potential change. It's always going to go to positive 30 and then back down to about negative 80 and then back to rest. So all action potentials will be identical, even if we have a stronger stimulus. So if we have a bigger graded potential, it will still generate the same size, the same duration action potential. All right. And this is important because as we think about how we can send different types of signals in the nervous system. So let's say you had a small sensory change, right? How would your nervous system distinguish that from a larger sensory change? And what does that is the is the frequency of the action potentials. All right, let me say that in another way. If I wanted to distinguish between a small change and a large change, if I wanted to lift a small load, right, and send a signal for a small muscle contraction versus a large muscle contraction, the way that the nervous system distinguishes or grades different types of signals is by the frequency, how many of those action potentials are generated. But in terms of an individual action potential, it's always going to be the same and it's all or nothing. So once that threshold value is met, action potential will occur. It will always go to positive 30, and then it will always go back to rest. So here's just an illustration of that. So here's one graded potential, a slightly larger graded potential, not enough to get us to threshold. Here is a third graded potential, which is large enough to get to that threshold. And so we generate an action potential. And then if you look at the size of the stimulus on the curve at the bottom, right on the graph at the bottom, here's the difference between this stimulus and then a supra-threshold stimulus, which is a larger change in the membrane potential that's going to be bigger than negative 55. Let's say it's going to go to negative 30, right? That is still going to generate the exact same action potential. So there's no difference in the duration, in the amplitude, in the size of the action potential, even when the stimulus is significantly larger. Okay. And then here's a link to that video. Again, please feel free to have a watch of this. This is also linked in Brightspace for you. Let's look at this in terms of a flow chart. Let's look at this in terms of an algorithm and kind of determine how this happens step by step. So this is just a different way to appreciate the events of the action potential. So we start with our threshold stimulus. And again, this must get to negative 55 in order for the next sequence of events to occur. Then we depolarize the membrane. And when we think about depolarizing the membrane, we want to understand the details behind that. It's basically the opening up of our sodium channels, right? Our voltage gated sodium channels, they get triggered off at negative 55. They open up, sodium rushes into the cell, which allows more positive charge inside the cell, and so that depolarizes the membrane, which is basically that opening up of sodium channels, right? And in terms of the permeability, so a different way to express this is that the permeability for sodium is going to increase as more sodium channels open up and sodium can move into the cell. All right, now we are thinking about depolarization as being a positive feedback example. So as more positive charge gets inside the cell, this is moving us away from negative 70. Negative 70 is our comfort zone. It's that resting spot, right? It's the set point. So as positive charge enters the cell, the environment inside the cell is moving further and further away from that negative 70, which is our set point. So we can think about this as being a positive feedback example. Not only that, as positive charge enters the cell, it further potentiates the depolarization of the membrane by allowing more neighboring cells to get to their threshold value, and then more of those sodium channels can open up. So this is just going to further depolarize the membrane potential, and this is really what results in the action potential propagating or traveling down the length of the axon. All right, now also happening when we get to that opening up of voltage gated channels is the 1 millisecond delay. So whenever we trigger at negative 55, that is, we trigger the opening up of sodium channels, we also set off that 1 millisecond timer where the inactivation gates are going to close after 1 millisecond of time. So then the inactivation gates on sodium close after that duration. And then the membrane permeability for sodium is going to start moving in the opposite direction. There should be a down arrow here. I would put it in, but it's going to just disappear on the next slide. But put a down arrow next to this membrane permeability box, because then the membrane permeability for sodium will decrease as those inactivation gates close. All right, so there's less sodium flowing into the cell as these gates begin to close. So the membrane permeability for sodium goes down, less positive charge is inside the cell. And so this begins to turn around the membrane potential, right? As we see less positive charge come into the cell, that begins to move in the opposite direction. Simultaneously, at that negative 55 threshold, there's another 1 millisecond timer that also goes off. That is the timer for the potassium channels. And notice that this is happening. So the 1 millisecond timer on the inactivation gate is syncing up with the 1 millisecond timer on the potassium gate. That is so that these two events can be timed to perfection, right? These two events can occur at the exact same time. This is what results in that spike, right? That point in the curve on the action potential graph is because at the exact same time, inactivation gates close, potassium channels open up, and they're perfectly synced to happen at the exact same time. So then that will further change the membrane potential as more positive charge leaves the cell because the potassium channels are now opened up. So less sodium is coming in because we've closed inactivation gates. More potassium is going out because we've opened potassium channels, both occurring at the exact same time. And now the membrane permeability for potassium begins to rise. Again, an arrow is missing here, so include that in your version of the notes, just so that it's clear. So this should be going up at this point as more potassium is moving, and then this one should have been going down. All right, so membrane permeability for potassium increases, potassium begins to leave the cell. Remember, potassium is moving in the opposite direction because of its outward ECDF. Potassium moves out of the cell, which is going to allow the cell to continue that downward slope where it becomes more negative inside, and then we approach sort of our comfort zone, which is down at negative 70. And so this will be an example of negative feedback. So as potassium leaves, less sodium enters, the inside of the cell begins to get more negative again, and that's going to get us back closer to where we were at the start at rest, which is -70. All right. Okay, so here is, again, a different way to represent this. I'm not going to go over this drawing in detail. I will share this as a PDF with today's recording, but in the interest of time, I'm going to skip over this. But definitely as homework, just practice drawing this out. How we go from graded potentials to depolarization, what's happening with depolarization, how the permeabilities are changing, what the gates are doing, right? The activation versus the inactivation. And then repolarization and how that is more potassium involved. And then hyperpolarization and what's happening here. Okay, I will explain IPSP and EPSP. I don't think we've introduced these terms yet. But these are examples of different types of graded potentials. And we will eventually talk about these two terms. But these are just ways that we can get to -55, which is threshold. All right, so just a bit of homework for you guys to practice drawing this out and just to use that as example to understand the action potential. Again, another study tool here. So this one is a really nice table showing you the different phases of the action potential, what the membrane potential looks like for each of these, what the voltage gated sodium channels are doing, again, what each of the gates on that channel are doing respectively, how sodium is moving, how potassium is moving, etc. So as a way to study this, try to clear out the table. So just block these tables out and then try to predict what would be in each of these boxes. And again, try to do this from understanding and not necessarily from just memorization. All right. Okay. Again, another really helpful study table. This one is looking at the graded potential versus the action potential and how they differ in terms of the strength, right? In terms of the ability for summation, in terms of the absolute refractory period versus the relative, right? And in terms of what ions are moving. Okay, so this is another example of a really good study tool. Try to block these out and then try to fill them in as a way to recall. All right. Okay, let us, I'm going to skip over these checkpoint questions. I will come back to these, but in the interest of time, I'm going to skip over these and move to lecture objective 3, which is the refractory periods, okay? But have a look at those checkpoint questions. I believe you have them on your slides, and then write some responses down, and then we'll review them when we meet on Monday. Okay, so lecture objective 3 is to describe refractory periods, including what causes the absolute and relative refractory periods, and explain their physiological significance. So why do we have these periods where we can no longer generate another action potential? So what is a refractory period? It is basically allowing the action potential to stop, right? It's the cessation of the action potential. And furthermore, it's the inability to reproduce another action potential in the periods that immediately follow the action potential that's currently occurring. Okay, so here's our action potential, -55, then we go up to +30, and then we come back down. We overshoot the membrane, and then we come back to rest, right? We overshoot the resting membrane potential, that is. So notice that there's a period during the action potential where we cannot stimulate another action potential. This is called the absolute refractory period. This is happening in the middle of the action potential. And this is because the sodium channels are inactive. So remember, at +30, we have not necessarily at +30, but triggered at -55. After that 1 millisecond, the inactivation gates close. This is why the inactivation gates are so important. So they inactivate the channels. So the channels are not only closed, but they're inactive. And they're not possible, it's not possible to open them, even if we had another graded potential that was stronger than -55. Okay, so in the absolute refractory period, it's not possible to have another action potential because the inactivation gates are closed. And even if a strong enough graded potential came along, it was not possible for more sodium to enter the cell in this state. The relative refractory period is going to be after the period of repolarization. So after we get back down to -70 and we go slightly more negative than the resting membrane potential, here we still have a lot of potassium channels that are still open. And because there's a lot of potassium channels that are still open, there's still some potassium that is leaving the cell. And because there's potassium leaving the cell, it's going to be harder to fire another action potential. Not impossible, but harder. Because more of that positive charge is leaving, the inside of the cell is beginning to get more negative. You can see that down here. So the membrane potential is even more negative than -70. So it's like we're starting from a more negative place than the resting membrane potential that we would typically start from. So we're going to need a really strong graded potential to overcome this negative difference and to get us closer to -55. All right, so it's impossible to fire another action potential in the absolute refractory period, but it's possible in the relative period, but it's just harder. It's just harder because potassium is still leaving the cell. All of the potassium channels haven't fully closed, which is representative of the membrane potential being more negative than -70. And if you think back to potassium's equilibrium potential, that would make sense, because potassium's equilibrium potential is way down here. It's -94. So until all of those additional potassium channels close, the membrane potential is going to be a little bit more negative than where it typically is. It's going to be closer to potassium's equilibrium potential. And so when we finally get all of those closed, then the membrane potential will get back to -70, which is that comfort spot. And then it's going to be easier to fire that action potential because we're starting from -70 and then trying to go to -55. But if we try to go to -55 from -80, that's going to require a bigger graded potential. All right, so again, looking at the absolute refractory period, it spans the entirety of depolarization and repolarization. So this entire period here, immediately surrounding the events of the action potential, the sodium inactivation gates are closed, and so it's impossible for another action potential to occur. For the relative refractory period, it spans the last phase of repolarization going into hyperpolarization. So notice here, if we look at the permeability for potassium, this blue curve, we can see that the permeability is going down, but it's not all the way to 0. So some of those voltage-gated potassium channels are still open. And so it's going to be harder to get to another -55 in this state of the channel until all of these voltage-gated potassium channels close.